This video is going to talk about the process of transduction. So if you remember, transduction is one of the three horizontal gene transfer mechanisms that bacteria use to acquire new pieces of DNA. So the shortest definition of transduction is viral mediated DNA transfer. So what that means is there's a virus, so in this case, more specifically a bacteriophage. So these are viruses that infect bacteria, is going to be responsible for transporting and introducing DNA to another bacterial cell. So what we have here is a bacterial cell, and this of course is not to scale because it looks like the virus is basically a similar size, so this would be much smaller, but this is the virus. And if you remember, what happens is all viruses have to use a host cell to make more viruses. So in this case, bacteriophage use bacterial cells. So in this case, this is the DNA molecule that exists in the bacterial cell, the host cell's DNA. And what the virus is going to do is inject its DNA into the host cell. So if you think about this, there are now currently two different instruction manuals. There's the bacterial DNA, but there's also the viral DNA. And so in many bacteriophage, one important instruction that is provided within the viral DNA is to actually generate some enzymes that destroy the host or the bacterial DNA. So rather than remaining an intact instruction manual, it's going to get broken down into little pieces. So it's like shredding the instruction manual. So now the only intact instruction manual is the viral genome. And what that's going to do is it's going to tell the cell to make all of the parts needed to synthesize more viral particles, so more bacteriophage. And so these are not assembled yet, but it's got all the pieces. It's got the viral DNA shown here in yellow. It's got the viral capsid shown here as this kind of little polyhedral shape is what we call it. And so all the pieces are there. So that's the synthesis part. And then, of course, what happens is assembly. So the bacteriophage parts are going to come together to form these new pieces. And so most of these bacteriophage, when they package them up, actually put viral DNA inside of it so that you'll get a continuation, and you'll generate bacteriophage that look just like what started this whole process. So that's what the virus is trying to do. But we want to look at this one that I've highlighted in red. That's known as the transducing phage. And so what's different about it compared to all the other phage around it is when that was assembled, instead of putting viral DNA inside of this capsid, there's going to be some piece or pieces of the shredded bacterial DNA. So those are shown here in red, right? So if you back up, the bacterial DNA has been shredded, it's been degraded, and so there are pieces of it. And so it is possible for some of those to get packaged inside of the virus when it's assembled. And so what that then leads to is, during the process of how this virus operates, the cell is actually going to lyse. And all of those newly assembled bacteriophage are going to be released. And so each of these appropriately assembled viruses, the bacteriophage, will go and repeat this process. But this bacteriophage, when it goes and attaches, it's not going to inject viral DNA. Instead, it's going to inject bacterial DNA. And so that's how a piece or pieces of bacterial DNA can be transferred to another recipient bacterial cell. And just as we've seen in other examples of horizontal gene transfer, it's not simply the transfer that's important. That's one important step. So how does the DNA get to a recipient cell? But then there's that second step of recombination. So the actual integration of the bacterial DNA into the recipient cell host chromosome.